That well, you're presenting a tool, at least in part, right, Jessica?
Yeah.
Awesome. That's one of those things, or if you can get it out there and people want to use it, it's very exciting. Oops. There we go. Whenever I start the YouTube, it's like 20-second lag, and then I hear my voice from 20 seconds ago, and I'm like, oh God. Put that away. All right, looks like we are streaming. All right, we'll give people just a couple minutes to check in. Laurel, how's everything out in Boulder?
It's good.
Yeah.
Crazy life.
Does it really look like that? Like your picture in the background?
Yeah, it really does.
Oh,
I mean,
awesome.
I'm not at work, I'm at home, but I can see like the Rocky Mountains from my office window. It's not the worst, I gotta say. Growing up in the Midwest, which I also love. It's a lot flatter, but yeah, it's pretty remarkable when you can like see 14ers from your window.
Oh, that's awesome. Are you from Madison or Wisconsin?
Yeah, from Wisconsin.
I love Madison. I think it's such a wonderful city.
Yeah, I love it too. I miss it. We haven't been back. Well, I haven't been back since like December 2019 because we haven't been traveling. So yeah, hopefully maybe this summer we can go back. I don't know. It's crazy.
Have you not been able to see your family in a long time?
No, my family's here all the time. They come over, which is great. We had a baby in April of 2020, and so we just haven't been traveling. So people come here instead, which is it's great. We don't have to go anywhere, and they can come to us. But yeah,
your kid's gonna grow up with a lot of stories about their
pandemic.
first couple years of life.
Oh yes, yeah. Pandemic babyness is real, so it's pretty funny.
It might be normal for them, I guess.
Yeah. Yeah, I hope I make it through. He actually caught a cold, which I don't know how that's possible. We don't go anywhere or do anything. He like stays home all day. But so we're all like sort of getting sick, which is really unfortunate. But I think I'll be okay for the seminar without like coughing or anything.
Well, if you do, it's okay if you cough on us because we're over Zoom.
Well, I'm tested.
Yeah, I mean,
it's definitely cold.
I think you're experiencing the thing that parents experience where their kid gets sick and everybody gets sick. It's just not from daycare. It's from
right. Like we haven't had this. He's 18 months old and it's like he's never been sick before. It's kind of insane, but
that's well, you know what? That's a blessing.
Yes.
Awesome. Okay. Since we have two people, we should probably start on time-ish, which is never my forte. So I will go ahead and introduce you. So welcome, everybody. Today, we are delighted to have two speakers. First up, we've got Dr. Laurel. Now, is it Hind or Hind? I should have asked you ahead of time.
Yep. Hind.
Dr. Laurel Hind. Laurel got her bachelor's from the University of Wisconsin-Madison in chemical and biological engineering. She went on to get her PhD from the University of Pennsylvania in bioengineering, working in the lab of Dr. Daniel Hammer. From there, she actually went back to Wisconsin to do a postdoc with Dr. Anna Huttenlocher, which some of you might remember we had on the seminar, I don't know, over a year ago. And then recently she joined the faculty at the University of Colorado at Boulder. And there her group uses microfluidic models of the infectious microenvironment to investigate how multicellular interactions, the physical environment, and soluble signals drive immune cell recruitment to an infection. So we're really excited to have her kick off today's seminar, and take it away, Laurel, the floor is yours.
Thanks. So I just want to start by thanking the organizers for having me. This has been an amazing seminar series. I've really enjoyed it the whole time it's been running. I hope it keeps going. And so I'm really excited to participate and to show you some of our work today. And so as Jen mentioned, we're really interested in looking at the neutrophil response to infection and more broadly, the innate immune response to infection. So if we think about sort of what an innate immune response to infection looks like, at steady state or at homeostasis before an infection begins, you have neutrophils circulating throughout your bloodstream in an inactive state, and you often have, or you definitely have tissue resident cells out in your tissues, such as macrophages, which are another innate immune cell we're interested in. And then following some sort of inflammatory event, in this case, we're really interested in infection, The pathogen itself, in addition to these tissue resident cells, such as macrophages, activate your vascular endothelium. And this causes the vascular endothelium to upregulate a number of pro-inflammatory signals, including soluble signals and adhesion receptors, which then activate your neutrophils, causing them to undergo this leukocyte adhesion cascade where they bind to the endothelium, roll along the endothelium, crawl, and then eventually extravasate through the endothelium. It's then known that these neutrophils can release their own signals, which recruit more neutrophils to the site of infection and recruit other innate immune cells, and eventually induce the adaptive immune system. And so we're really interested in how all these different cellular interactions play into the process. So as you might have noticed, there's a lot of cellular signaling happening between the endothelium and the neutrophils and the macrophages and other tissue resident cells. And so we really want to investigate how these interactions drive or enhance the neutrophil response. And so today I'm going to tell you a couple of stories. The first two stories are really the result of the work I performed as a postdoc in Anna Huttenmacher's lab at the University of Wisconsin-Madison, where we sought to develop a system to look at these multicellular interactions in the context of infection. So looking at how neutrophils are recruited to infection and how these multicellular interactions play a role. And then by giving you a little bit of a teaser into what we're looking at in my lab currently. And so I'm going to start talking about our development of this infection on a chip system. And so the neutrophil response to infection is often carried out using one of two very common experimental models. So there are in vitro models that have many advantages. These often allow you to use human cells, primary human immune cells, which are great because, well, cell lines exist for immune cells, primary where it's at. They're the most relevant cell type to be looking at. These type of models give you a lot of control over the type of signals that you add to your system, so you can investigate the neutrophil response to very specific pro-inflammatory cues, or in some cases using like this transwell assay, look at individual cell-cell interactions between a neutrophil and an endothelial cell, for instance, and how that would affect neutrophil migration. However, they lack a lot of complexity that you might want in a system where you're trying to mimic an in vivo response. And so, as many of you know, and I'm sure many of you use, in vivo models can be really invaluable for looking at these type of responses. So when we're talking about innate immunity, animal models that are very common are mouse models and the zebrafish model, which Anna's lab uses quite a bit. To look at the innate immune response. And these models can be really great because they have that inherent complexity where you have multicellular interactions and full animal structures, tissue structures. But because of this complexity, it can be hard to tease apart exactly what's happening in a cell-cell interaction. And so the goal of my lab and where I started in Anna's lab was to sort of pull advantages from these two different systems into what we call a biologically inspired in vitro model. So these are microfluidic models that try to recapitulate some aspects of the in vivo environment, like having a model endothelial blood vessel and an extracellular matrix, while preserving some of the tunability and functionality of an in vitro model and allowing you to use primary immune cells. And so in designing our first in vitro biologically inspired in vitro model, we wanted to pull in as much of this neutrophil response as we could. And so the first thing we were interested in looking at was the neutrophil interaction with the vasculature. And so we used this system where we could develop, like I said, a model endothelial blood vessel. And so in this case, we have lumen in a collagen extracellular matrix that is coated with endothelial cells to represent a blood vessel. And then we were able to seed in primary human neutrophils and use a source of live pathogen. So in this first story, we used a bacterial pathogen, Pseudomonas aeruginosa, and look at the neutrophil response. And the way that we did this is using a Luminex device, which was developed by Dave Beebe's lab at UW. So Dave and Anna have a long-standing collaboration. And so this was a really great crossover between the two labs. And in this device, there's this PDMS well that has a PDMS rod that runs through it. And you can then polymerize a hydrogel. So in our case, as I said, we're using collagen 1 as an ECM mimic. And then you can remove the rod. And what you're left with is this hydrogel that has this hollow lumen core. And this is what it looks like using SHG imaging, where you can see the collagen in the matrix and this core in the middle. We then were able to seed endothelial cells. And for these projects, we were using iPSC-derived endothelial cells. And what was great about them is when you seeded them in the lumens, they created these patent lumen vessels that surrounded the entire lumen structure of the collagen. And they also formed these monolayers that were aligned in the longitudinal direction, much like you would see in vivo. And so, as I mentioned, the first thing we were really interested in asking was, how does the endothelial vessel influence neutrophil migration? And in order to answer this question, we created two types of devices. So we created devices without an endothelium here on the left. And so in this case, we created that collagen matrix with the hollow lumen. We never seeded endothelial cells. And then we created devices with an endothelium. And in both cases, we added Pseudomonas to that top source port for our bacterial pathogen and watched the neutrophil response. And what you can see as the video plays is that we saw this really dramatic response when we had an endothelium present. And so we saw enhanced migration and recruitment in the presence of an endothelium. And so we went on to quantify this and found that both the number of neutrophils that left the lumen, so the number of extravasated neutrophils, and the distance they were able to travel away from the lumen were increased in the presence of the endothelium, showing this enhanced recruitment that occurs when you have this endothelial vessel. The other thing you might have noticed from this video is that the neutrophils in the endothelium case seem to migrate for a really long time compared to those without an endothelium. So on the left, the neutrophils start to migrate out, but they kind of peter out and stop migrating about 480 minutes in, maybe 500 minutes in, which is about 4 hours. And this is what you might expect if you ever work with primary human neutrophils. It's sort of known that they don't survive outside the body for a very long time, and you have to experiment into those first few hours. But in our case, when we added this endothelium in, we saw this much longer neutrophil migration. And so to confirm that this was actually a survival enhancement, we did a live dead assay using propydium iodide to stain for dead cells, and found that, in fact, after about 4 hours, we started to see in this unlined, no endothelium case, so the only cells in here are neutrophils, that we had really bright staining. We had a lot of cells dying about 4 hours into our experiments. Whereas in the presence of the endothelium, we had almost no cell death. And when we quantified this, we saw that this held. So without an endothelium, we have significant cell death even after 4 hours, which continues through our experimental window of 16 hours. But with the endothelium, we saw only about 10% of the cells dying even within that first 16 hours, which is really remarkable. And you can notice here that there's really a lack of propydium iodide staining out here in the periphery. And so this led us to, of course, ask, what signals are being produced that could be leading to this enhanced recruitment and enhanced survival of the neutrophils when we have Pseudomonas and this endothelial vessel? And so to look at this, we used a multiplex ELISA system where we collected supernatant from vessels that did not have a bacteria, only had the endothelium, and used this as our baseline for secreted protein analysis. We then added the Pseudomonas in and looked for signals that were altered in the presence of infection. And we saw two really interesting candidate molecules, GM-CSF and IL-6. And importantly, both of those were still present when neutrophils were added into the system, and they were still upregulated. And so these were the first two that we really wanted to explore. GM-CSF is a known pro-survival signal for neutrophils. And so we first looked at how GM-CSF signaling might be influencing the neutrophil response. And to do this, we used a blocking antibody against GM-CSF to block the signaling. And what we saw is that there was not a significant difference in the migration of neutrophils in the presence of this GM-CSF blocking antibody. And so a number of neutrophils were still able to leave the lumen, and they migrated a significant distance, but it really plateaued. And that was due to the fact that we actually did see a survival benefit with GM-CSF. So in the case where we had our blocking antibody, we saw significantly more cell death, indicating that GM-CSF was enhancing the survival or lifetime of these neutrophils. But this didn't really answer the question of what was leading to this enhanced migration that we saw. And so we turned back to IL-6. IL-6 is a known pro-inflammatory cytokine, although it's not really known to be a chemoattractant. It is known to stimulate an inflammatory state. And so to look at this, we again turned to a blocking antibody. And in this case, we blocked the IL-6 receptor. And what we saw was that in the case where IL-6 was blocked, or the receptor signaling was blocked, we saw this extremely reduced migration, where only a few neutrophils are able to leave the endothelium, and we really have reduced neutrophil migration. And when we quantified this, we saw that both the number of neutrophils outside the lumen and the distance that they were able to travel away from the lumen were reduced in the presence of this IL-6 receptor blocking antibody. And interestingly, when we looked at cell lifetime, there was really no effect of blocking IL-6 on cell lifetime, indicating that IL-6 and GM-CSF were really playing distinct roles in our system. And so sort of to conclude this little story, we showed that the endothelium is really important in enhancing neutrophil migration to at least the bacteria Pseudomonas aeruginosa, and that this was mediated through secretion by the endothelium of both IL-6 and GM-CSF, which led to increased recruitment and lifetime respectively. The other really interesting thing that this showed us was it highlighted the importance of using these biologically inspired models to look at neutrophil recruitment, especially in the context of infection. So we were able to see how this endothelial cell neutrophil paracrine signaling really drove the neutrophil response, which we wouldn't have been able to see necessarily in a more simplified system. And so this was a great sort of proof of concept study. We were able to see some really interesting things in terms of IL-6 and GM-CSF signaling, but it's really only one infection. And so we wanted to expand our studies and really investigate how paracrine signals and how the system might work when looking at other types of infection. And so for our second story, we turned to a very different type of infection, a fungal infection with the fungus Aspergillus fumigatus. And we really wanted to know how would the neutrophil response differ in a fungal infection compared to what we'd seen in a bacterial infection. And so the first thing was to just see if the neutrophils would even respond to the fungus, if the fungus would create an infectious source in our device. And so in this case, the neutrophils are labeled down here in green. And when we play the video, we'll see the hyphae begin to form in purple. And as the hyphae form, you can see that there's sort of this initial patrolling neutrophil migration. And then once the hyphae become visible and come close to the lumen, the neutrophils accelerate out. And this is seen when we quantify neutrophil migration. So in contrast to the bacterial migration, where we started to see sort of this fast acceleration that then plateaued as the infection proceeded, here we saw this sort of lower level of patrolling migration, and about 4 hours post-germination, which is how we lined up these experiments, we then saw this acceleration of neutrophil migration and neutrophils really migrating out. So the other thing that you might notice is in this case, the neutrophils are starting to coat the hyphae, and they coat that first hyphae that sort of appears on the screen. And we saw this over and over. And so this was very reminiscent of what is known of a neutrophil swarm. And so in neutrophil swarming, you get these large clusters of neutrophil coming together. And this can happen in bacterial infections and fungal infections. But it was really interesting that we could see this in our device. And one reason we were really sort of amazed by this is because it so closely recapitulated what we were seeing in vivo. So the images on the right are from another former postdoc from Anna's lab, Emily Rosowski, who's now at Clemson University as an assistant professor. And she was looking at Aspergillus infection in the zebrafish model. And so on the right here, she's using an RF8 null mouse, which has depleted macrophages. So this is really a neutrophil response to Aspergillus, which is much like what we have in our device. And when she looked at these, she used a Sudan black stain to stain for neutrophils and saw these huge neutrophil clouds that were surrounding the Aspergillus hyphae. And they looked a lot like what we were seeing in our device, indicating that much of what we were seeing was potentially mimicking what you would see in vivo. And so, as some members of our audience might know, neutrophil swarming is driven by LTB4 signaling. And so it's the signal relay pathway. And so the first neutrophils that arrive at a site of inflammation, or in this case, infection, can undergo the signal amplification, where they then release a secondary gradient themselves, which leads to an amplification of the signal and this accelerated neutrophil swarming. And many groups have studied this, including Tim Lemmerman's, Carol Perrin's group, and have looked at how this signaling occurs and have found that LTB4 is the primary mediator, and that it is released by neutrophils and then taken up by trailing neutrophils to lead to the signaling. And so to determine if this was what was happening in our system, we used a chemical inhibitor against this 5-LO molecule, which is important in the generation of this LTB4 gradient. And what we saw was, in fact, when we blocked this secondary gradient and this accelerated neutrophil swarming, we got reduced neutrophil migration. And so we didn't see that same acceleration that we saw with the presence of LTB4 signaling. We also saw a significant reduction in the number of devices that had swarms. And so this really indicated that this paracrine neutrophil signaling was really important for the response we were seeing in our device. This led us to ask other innate immune cells might also contribute to the neutrophil response to Aspergillus. And so thinking about our device and what it models and what might be a relevant cell type to look at, we turn to monocytes. And so monocytes are another innate immune cell that are present in the bloodstream. And so seeding them in the device with the neutrophils was a relevant way to study them. And we really wanted to know, do they alter the neutrophil response? And so what we saw was that when we added monocytes, we did see a really enhanced neutrophil migration. And so once again, we're showing that some sort of paracrine signaling between innate immune cells is enhancing our response, highlighting the importance of sort of studying these cell types together. And so to determine what might be driving this response, we again turn to our multiplex ELISA system to look at signals secreted by monocytes that might alter this response. And interestingly, there were two different signaling families that were significantly altered in the presence of monocytes. And so there was the MIP1 family and the IL-1 family. And in both cases, these are known pro-inflammatory molecules. And so we weren't really sure where to start. So what we did is just systematically go through and block signaling from each of these candidate molecules. And the only one that really showed a strong difference was MIP1 alpha. And so in the presence of MIP1 alpha, we saw a blocking antibody against MIP1 alpha, we saw a significantly reduced neutrophil migration, indicating that this paracrine signaling between neutrophils and monocytes might be mediated through this signal. And so this was a really interesting study because it showed us that our system could at least recapitulate some aspects of the in vivo response with this neutrophil swarming. And it really highlighted again how important it is to look at these paracrine signals. And so these two different stories have really built the research program that we're now undertaking in my lab. As a caveat, I started my lab in January of 2020. So as you might imagine, it's a pretty hard time to be a new professor. And so our research is really sort of just getting off the ground. So in the last just 2 minutes here, probably, I'm gonna give you a little teaser of what we're doing, but most of what we have, there's very little analysis or interpretation because we just don't have enough yet. And some of it is even single replicates because it's very preliminary data. And so there's sort of three main thrusts that we're looking at. We are continuing to follow up, looking at different multicellular interactions in neutrophil function. This is being headed by one of my first graduate students, Isaac, who's looking at how other vascular cells, such as pericytes, might be regulating the neutrophil response and how different pathogen sources might lead to different responses. In this realm, my master's student, Tanisha, is also looking at how macrophage neutrophil interactions affect the neutrophil response in our device. Our second project is looking at how the physical environment, specifically how ECM composition and concentration is driving this response. And this is led by my graduate student, Chris. And then finally, we have a section of our lab looking at how these more rare innate immune cells, such as myeloid-derived suppressor cells, function in response to infection. And this is being led by my student, Hannah. So just 2 or 3 slides on some data. In terms of the multicellular interactions, we've started to develop these pericyte lumens. And so our goal is to eventually have endothelial pericyte multicellular lumens. But right now we're just looking at the pericytes themselves and getting this optimized. We have seen that pericytes are able to stimulate neutrophil migration to a lesser extent than endothelial cells, but this is kind of in line with what's in the literature, so that's pretty exciting. In terms of looking at the varying pathogen sources, we're also looking at a range of bacteria from extracellular, intracellular, gram-negative, and gram-positive. And what's been really interesting is we've actually seen this extremely robust neutrophil response to Listeria in our devices, much higher than our sort of baseline Pseudomonas, which is down here in red. And so we're now digging into why that might be occurring. For the macrophage interactions, we're actually using a THP1 monocytic cell line and seeding them in our devices and then differentiating them in situ so that we can create our lumens and look at neutrophil migration with macrophages in that sort of interstitial space. And so Tanisha confirmed that over our 2-day differentiation period and then the 4 days required to make our lumens, we have really good cell viability and a highly differentiated population of macrophages. And what's really interesting is, so this is a single day's experiment, so just hold that in mind, but over 3 devices looking at neutrophil migration in our 2 different situations, we are seeing reduced neutrophil migration when macrophages are present, which might mimic some of what's known about macrophages' role in potentially driving reverse migration of neutrophils or limiting their response. And then finally, our ECM density project, we're looking at how different collagen densities affect neutrophil migration, both directly because of their increased stiffness and reduced pore size, but also potentially by changing how the endothelium reacts. And so we've been looking at devices that have low density 2 mg per mil collagen versus high density 4 mg per mil collagen. And we actually see the significant increase in migration in the more dense ECM, which is really interesting. And this holds in both the endothelial case and the no endothelial case. Although again, like we saw before, we're seeing that without an endothelium, we have significantly reduced migration compared to when we have that endothelium. So there's still definitely some interplay happening there. And so that's just a little teaser of things that we're looking at. Not much there yet, but things to watch for the next few years. And so with that, I'd just like to thank my lab. So this is my lab on the right with all our undergrad, my 4 graduate students, my postdoc advisor, Anna, and our collaborators in the BB lab. And just a quick shameless plug, we're always looking for talented graduate students and especially postdocs. So if this seems interesting to you, feel free to check out our lab website and our positions. This is actually a photo that I took from the parking lot of our building. So it is a pretty beautiful place to work. It's not always this snowy, I promise. We get much less snow than I used to get in Wisconsin. So it's not that wintry, but it's pretty gorgeous. And with that, I'm happy to take any questions.
Awesome. Fantastic. Thank you so much, Laurel. And I just want to say, wow, that you've started a lab and had a baby all since starting at the beginning of the pandemic. And that's really impressive. And so it's awesome that you guys are getting things off the ground in your new lab. That's great. So I have a couple questions, but we'll see if we can get to them. First from Rachel by Lyra, wondering about, could you perform the assay with HL60s?
Sorry, you cut out for just a second. Could I perform the assay with what?
Yeah, from the first part of your talk, HL60s.
Yeah, that's a good question. Maybe. So I haven't used HL60s specifically. We've used the PLB985s, which are a derivative of the HL60s. and seen some success. They don't migrate nearly as well as primary human neutrophils, which I think holds with most in vitro studies that look at them. I actually do have a grant to work on looking at POBs in this device, so we're hoping to get them up and running. But yeah.
Thank you so much, Laurel. Next question is from Hava. She's asking, is IL-6 a chemorepellent? Does it form any sort of gradient?
That's a good question. I don't think it's a chemorepellent. My basic understanding of IL-6, and I will again provide the caveat that I'm an engineer, not an immunologist. I just did an immunology postdoc, so I pretend to be an immunologist. But it's a pro-inflammatory cytokine. So my guess is that what it's actually doing is just promoting random migration. And so it's sort of activating cells in a way that they're becoming more migratory and more interested in sort of seeking out other chemoattractant signals. We did look at IL-6 sort of in a very basic chemotaxis assay to see if we could stimulate chemotaxis with it, and we didn't really see that, which again, I think is in line with what the literature would say. So my guess is that it's really more of just a sort of pro-inflammatory, getting the neutrophils excited sort of signal.
Fantastic. Taehyun Kim is wondering, do you check the polarity of the macrophages? Are they more M1-like or M2-like?
That's a great question. So right now, not yet, because this is such a new project, but it's definitely something we want to look into. And actually, I want to check the polarity of what we have, but then also try to drive polarity and see if we see a difference, if we can differentiate them towards an M1 or M2 phenotype. Our goal is also to get primary human macrophages into the device. They're just trickier. And so for right now, we started with THP1s. But yeah, I'm really interested in macrophage polarity. I did my entire PhD on macrophages and looking at their migration. And so that's something that we definitely want to look at in the future.
Awesome. So next, Abhijit says, great work as always. Do you think that increased survival and motility of the neutrophils and the presence of endothelium has to do with neutrophil endothelial interaction only, or does endothelium respond to the infection to promote the survival and motility for neutrophils?
Yeah, so I think it's an infection. So, so we did the control where we did infection and no infection, and we saw very little response from the endothelial cells in terms of secretion. So when you don't have the presence of the Pseudomonas, the endothelial cells don't secrete much, and the neutrophils don't get out. And so because of that, I think it's definitely in response to the endothelium, in response to the infection. And we are in our new project looking at the different pathogens, looking at maybe how different pathogens differentially cause endothelial cells to secrete different pro-inflammatory molecules, to see if that might be one way to look at treating different infections.
Just as a quick follow-up to that, is there any spatial response of the pathogens? Is there any gradient? Do they glom on to the endothelium? Is there something that you can see in their behavior that they're responding?
Yeah, that's a great question. So in the Pseudomonas case, we did look at a tagged bacteria, so it was fluorescent, and it initially forms a gradient, but it fills up the whole device outside of the lumen very quickly within, I want to say 30 to 40 minutes. And so, but it's excluded from the lumen. So Pseudomonas is an extracellular pathogen, and so it doesn't actually get into the lumen. So another thing we're interested in looking at with the intracellular versus extracellular pathogens is if those intracellular pathogens actually get into the endothelial cells, and then maybe even into the lumen, and if that causes any differences in the neutrophil response, because that's just a totally different mechanism of infection. But in our device, they're pretty much ubiquitous. They very quickly sort of fill up the whole device.
Wonderful. There's one last question, and then maybe we will move on from Thomas Smith. How important is it to have tight endothelium, or would it be sufficient to just have endothelial cells homogeneously spread to obtain increased motility?
That's an interesting question. So we did test vascular permeability in these devices. And so just using a diffusion assay, we know that they have pretty good barrier function. In infected state. And then once you add the Pseudomonas in, you see this increased vascular permeability, which matches what you see in vivo. During an infection, your endothelial cells become more permeable. But we never did sort of a suboptimal monolayer where you have like big holes. So I'm not sure how important that neutrophil extravasation step where it actually squeezes through the endothelium is. I know other groups have looked at that extensively and seen huge differences in neutrophil activation after going through that step. So I would guess that it is really important, but we didn't directly test that in our device.
Thank you so much, Laurel. That was a wonderful talk. And if there are any more questions popping up later, we can maybe go in the end.
I'll be around.
With that, I would like to introduce now Dr. Jessica Henty-Ridilla. She is currently an assistant professor in biochemistry and molecular biology and neuroscience and physiology, at SUNY Upstate Medical University in New York. She got her PhD in plant biology from Dr. Christopher Stager's lab and moved on to Dr. Bruce Good's lab for her postdoc work, where she studied actin assembly and several proteins that coordinate dynamics of actin and microtubules. Her lab uses advanced microscopy and classic biochemistry to study actin and microtubule proteins that are regulated in normal and neurodegenerative diseases. Today, she's going to talk everything about prophyland, I'm guessing. So thank you so much for taking our invitation, and we're looking forward to your talk.
Okay, you can see the slides, right?
Yes, yeah.
Okay, good. Okay, so now we're going to talk about actin and microtubules, which are some of my favorite proteins ever, and prophyland, which is also up there. And we're going to talk about this tool that we sort of developed in the lab that's going to be really useful for us, I think, like as we move on in the future. Anyway, all right. So just to remind everyone, I know everyone probably knows this already, some basic actin and microtubule biochemistry for you, right? So actin, there's actin monomers and there's actin filaments. To make an actin filament, we have to take monomers, and it's really hard to make an actin filament just based on biochemical principles alone. But we have things that are called nucleation promoting factors like the ARP23 complex or formin or enivasp. There's actually a whole bunch of them that will actually sort of help us get over some of these rate limiting steps to actually make a filament. And bonus, sometimes we get special structures, either branched actin filaments versus like maybe straight filaments. Microtubules, you know, what's great about actin and microtubules is they're both polar molecules. That means that they have an end that grows faster and an end that grows slower. So for microtubules, they actually get assembled from dimers of tubulin. Now, if you wait long enough or you have a high enough concentration, this will just happen on its own. But usually what we do is we either make these little seeds from GMP, CPP, stabilized chunks of microtubules as a sort of template. Or in a cell, there's nucleation promoting factors like gamma-Turk that provide that little like seed for what the microtubule can like grow off from. And these are hollow tubes that are bigger. They have this extra bit. I mean, microtubules are just extra anyway. They're very dramatic. They have this extra property called dynamic instability, which basically means, hey, I'm a microtubule, I'm growing, I'm having a great time. Something happens and they're very dramatic, and they all of a sudden switch from this period of growth to rapid disassembly, which is actually called a catastrophe. So that's all you need to know. You don't have to worry about anything else. So this talk is about prophyland. Why do we need a tagged prophyland? Well, first of all, prophyland is involved in a lot of different cell processes. So you know what? Pick your favorite one that's on that list there. Prophyland probably does something with it. I'm not just saying that, but it's true. So prophyland actually is pretty cool. And there's a reason I just told you a little bit about actin and microtubules. So generally speaking, based on lots of work from lots of different model systems, people like to think of prophyland in a one-to-one complex with actin monomers. And when it's in this state, it can either promote nucleation with special nucleating promoting factors that have polyl-proline residues, like enivasp formins. It's sort of inhibitory for ARP complex mediated assembly, but that's not really because of ARP complex, but more because of the activators of the ARP complex. It actually inhibits the spontaneous assembly of actin filaments because it's in the same binding spot as where the new actin monomer wants to join. Now, one thing that happened that we discovered during my postdoc was prophyland could also bind to the sides of microtubules. We weren't the only person to see this. Roger Carlson's group saw this in cells very, very elegantly. And past work by Walter Ritke actually showed that prophyland 1 and 2 could bind to columns that had, that tubulin could bind to prophyland 1 or 2 biochemistry columns. So a reason we are interested in prophyland is we'd really like to dissect what's happening, what makes prophyland interact with actin or microtubules at any given time. And if we had a tool to do that in the test tube or in cells, that could be really beneficial to us. So here's the crystal structure of prophyland in blue, and this is sort of relaying some of the things that prophyland does. On one side of it, it binds to the actin. On the other side is where it does this polyl-proline business. So that's actually like really helpful. It can bind two things at the same time in that way. But it kind of prevents or presents some challenges for when you go to label it. So you can't really put a tag on one end or the other. The N and C terminus is actually here. It kind of interferes with this polyl-proline binding. Although people have, and many people have made fluorescent versions of prophyland. There's at least 10 different versions out there. The reason we went for more is because either prophyland's compromised in some way, it was in a different model system than humans. And so I guess the rules were just a little bit different. We tried all of the strategies and sometimes got some aggregation. Probably the best one that I've seen that's out there as far as both characterization and maybe lack of off-target things was this one also from Roger Carlson's group, where inside this loop that you can sort of see there, they inserted a citrine molecule. So it kind of hangs out there. And what was great about this was it could still bind polyl-proline just fine. But if you look in the methods and deep in the paper, some part of actin binding actin wasn't quite as functional. So this is really good. We wondered if we could do a little bit better. And this is just a zoom in to show you that there's like residues specifically used by people who've studied prophyland for separating prophyland's actin binding activities, that's R88, and also the polyl-proline binding activities, which is this Y60, and that's where they're located. Okay, so basically current probes compromise different parts of function. Okay, so what do we do? Well, first, if we have this, we can look at endogenous levels, what's happening to prophyland, which is pretty good because this protein's very abundant. We can see where it localizes because I don't know if anyone's ever looked at prophyland. Maybe it was just me. If you just have a GFP or a fluorescent version of prophyland, you're going to see your cell. You're going to see your cytoplasm. And, you could say that it localizes with everything, but that's probably not true. There's just a lot of prophyland there. So maybe this gives us an opportunity to figure out where it might localize, how it regulates actin and microtubules at the same time. And also because there's disease relevant mutations for cancer, but what I'm particularly interested in is actually amyotrophic lateral sclerosis, which is a neurodegenerative disease. And also part of my postdoc, we learned that certain mutations cause this microtubule binding activity to go away. So I'm really curious to see like what exactly prophyland's doing, particularly in the context of that disease. Okay, so here's some challenges. Prophyland is really small. It's smaller than most fluorescent probes, although we did put this up on bioRxiv and someone reviewed our work. So we had a really positive experience with like ASAP Bio. And they told us that there's actually this really cool small tag out there now. So, maybe that will be cool. We'll probably try that. Prophyland interacts with a lot of ligands, probably over 100. Traditional labeling approaches can be cytotoxic. So I could do things to label on all the cysteines. It causes some aggregation. I can mutate the 6 cysteines out, except for the one that is relevant for the disease. It still aggregates. And then if you tried to do that in cells, it would kill the cells. So you'd have to like inject it into cells. And That's a technique that I have not tried or mastered, but the people who do it, it's beautiful. And they should keep doing it because it's amazing. And then there's a lot of prophyland in a cell. And depending on what cell type you're using, it's somewhere between 20 and 500 micromolar. So it's a lot. Okay, so here's what we did. We took prophyland, we added a linker and tagged it with an M-Apple tag. Now, here's an overly honest method for you. The reason we used M-Apple is because we wanted something in that wavelength because our actin is green and our microtubules are a different color. It's not the greatest probe out there. There's probably better fluorescent probes, but what I hope you'll appreciate at the end of this is what's great about this is this linker. So we can interchange a different probe if we really want to have better fluorescent properties. So that's something to just keep in mind. And then we purified it. And so here's a gel, Kumasi gel. And on the bottom over here, I'm showing you our normal untagged prophyland. Our prophyland, when we purify it for our biochemical analyses, is not tagged. It's like hardcore old school biochem. Here's our M-Apple tagged prophyland. So it's at the size that we expect it. And there's nothing else that's like in there. Okay. Cool. And just as a fun aside, I also really like the color pink. So it's really fun when you purify this because it is bright pink. And when you're expressing it, the cultures look like it's Halloween or something. It's exciting. Okay, but is this functional? So the first thing we did, and by we, I went down the hall to someone who's an expert at this because I don't know how to do this. So we went down the hall to Patty Kane's lab, and her student Farzana did these liposome pelleting assays for us on 2 different kinds of lipids. We chose PIP2 and PIP452P. Prophyland loves to bind lipids normally, so we wanted to know that our prophyland could bind lipids, okay? Now, I'm showing you this little like cutout here, but what you should know if you look at these controls, we have some controls that have additional just liposomes in there. Prophyland loves to bind those too. So we had to subtract out that signal for these specific lipids that we're telling you, but I think that that's actually a good sign because it just loves binding lipids. And so we measured the specific binding of the different lipids for our untagged and our tagged version, and it looks pretty good. Okay. So then this is what I really want to know. So it's not here, but here's the question I really want to answer. So these ALS mutations, there's 8 of them. Does it do something to actin? That's actually like a very simple question that's very hard to answer. And so one of the things I wanted to do was to just know, does it bind actin differently? And there's a couple different ways you could do that. One that would be really helpful and useful is fluorescence anisotropy. Now, why hasn't anyone done this? Because this chart that you see right here is what happens when you use prophyland unlabeled with fluorescent actin for fluorescence anisotropy assay. If you look at that, that is not binding, right? You would say that is not binding. That did not work. And I think that the reason for this is prophyland is so small that you can't actually detect the change in the spin when you have the reactions with prophyland in there. So if I had a fluorescently tagged prophyland, it's going to be bigger, so we might be able to detect it better. And also I have a different probe that I can use. And usually if you have the smaller thing for anisotropy, you're going to get more of a difference to detect for detection. So another way you could do it is you could do this competitively. And one thing that we always forget, at least I always forget because I was in a plant lab before and then went to a lab that did a lot of stuff with yeast, is mammalian cells have this protein thymosin beta-4. And that protein loves to bind actin about 10-fold better than prophyland does. So we could do a competitive assay. So this is just to show you that in our hands, in this case, we're using the fluorescently labeled thymosin beta-4, so our actin's unlabeled. We can get a very nice binding curve with fluorescence anisotropy. Here's what happens if we do that competitive assay with the untagged prophyland as well as our M-Apple version. There it is. You'll notice that the KDs we get are around 100 nanomolar, which is really nice. They're not statistically different from each other. And it's in agreement with some ITC measurements that have been done by various labs for wild type prophyland. So that's very nice. And then we can also do it the other way where we have unlabeled actin and now our labeled version. And the KD is very similar if we perform the assay that way. So that's pretty good. We haven't done this for all of the ALS mutants yet. That's something that we're working on, but we're really excited that we have this tool for that. So there's that. Okay, so another thing you could do is you could do a pyrene assay, which overall measures sort of nucleation, or you could do total internal reflection fluorescence microscopy. So these are these cool biochemistry on a cover glass movies where we take purified actin that we've labeled and we flow in whatever protein we want to see what the effect is on actin. So over here, I'm showing you, here's our pyrene fluorescence of just actin by itself. Wild type prophyland inhibits that. That's what it does. There's our M-Apple version. And here's what the turf movies look like. So here's actin on its own, and then actin and prophyland. And you'll notice that in the reactions that contain prophyland, there's less overall actin filaments getting nucleated. Okay, you get the idea. Okay, so here's just a montage showing you that movie that I just did. So there's that. That looks pretty good. They're pretty equivalent. But what's great is we can actually quantify this. So we went and counted different fields of view and saw that there was very similar, not statistically different amounts of the numbers of filaments in the reactions that have wild type or are labeled prophyland. So it seems to be inhibiting actin assembly just fine. Okay, but what else could you do if you have a labeled prophyland? Now, on one hand, I was really excited about this, and on the other, don't get too excited, everyone, because it turned out to not be as cool as I thought it was going to be. So you can look at prophyland at the same time that you can look at the actin. And here's what happens when you do that. Okay, there's some little like pink prophyland spots in there, and there's an actin filament in there, and there's the merge. And if you look in that box, I'm showing you the one time where I thought maybe I'd see something cool. So there's a reason why I didn't see something cool. Prophyland is binding actin monomers, not filaments. For most stuff, there's work out there that says if you have high enough concentrations of prophyland, you might be able to start to see it associating with the growing end. Now, maybe in this one filament, this one example, we by no means tried to optimize this. We just wanted to say, here's what it looks like when we do that. We found this one example where maybe there's a prophyland associating with the growing end of a filament. That would require more work, but you could imagine that that could be really helpful for possibly like FRET or some cool assay there. Someone should do that. We're not going to do that, but someone who has that expertise, like I would love it if they would do that. That would be amazing. I think we could learn some cool things there. Anyway, so this is not too surprising because prophyland's binding monomers. So there's that. Okay, one other thing that prophyland does, in case you guys don't love formin as much as I do, or haven't thought about it in a while, formin's that blue thing, and yellow in this case is prophyland, and it's bringing in prophyland-bound actin monomers, and it does this this is what it does to accelerate actin filament assembly, okay? So that's probably what's happening, but I'm gonna show you a pyrene assay. This is different than the one I showed you before, but the same controls, okay? So prophyland or our M-Apple inhibits. That's what happens when you add formin. You get this really fast burst of actin assembly. Here's what happens when you add MDA and prophyland 1, the wild type. And there's what happens with our fluorescent tagged version. And what I'm showing you, the shaded area is 3 different pyrenes, and that's the standard deviation between them. So you're seeing like the average of that, which might be the best pyrene assay I've ever done, but just saying. Okay, here's what the turf looks like. So what you'll notice is there's a lot with actin and MDA, there's a lot of nucleation. So that field of view gets full really fast. Whenever prophyland is around in there, there's fewer filaments overall, but if you look very carefully, like at the very beginning, the reactions that have prophyland are, the actin filaments are longer, and that's because actin is actually like the elongation is getting accelerated. Okay, and then just, because sometimes movies don't work, there's the montages that helps if you can see that easier. So it seems like our tagged M-Apple prophyland can stimulate formin-mediated actin assembly just fine. And that would mean that it can bind poly-L-proline, but we did not directly test if it could do that, okay? And again, Here's what it looks like if you add these together. And I didn't even try to quantify this really, but like, you know, if formin's there, you might suspect that you might suddenly see more incidences of prophyland near the barbed end because of its interaction with formin, but we didn't really look at that carefully. And I do think that to really get at that, we have to like optimize exactly how much prophyland we have and how we're visualizing it, because there's still a lot to see at the concentrations that we use. Okay, so here's the crazy thing. So what about microtubules? So here's what happens. These are microtubules. On the left, I'm showing you microtubules just polymerizing, and then in the presence of prophyland or M-apple prophyland. And what we saw when I was a postdoc was in the presence of prophyland, microtubule assembly could be, the elongation rate went up, okay? And we saw also more microtubules present in some of the reactions that had prophyland compared to tubulin by itself. And if we titrated down to values of tubulin that shouldn't make microtubules because it's below the critical concentration, we could sometimes see microtubules polymerize in the reactions that had prophyland. And we didn't make a big deal about it because we didn't know if it was doing something to stimulate microtubule assembly. But with the labeled prophyland, we could now see, is something cool happening with microtubules? Like you might imagine if it's about elongation, that the growing end of microtubules might be enriched for prophyland or something, okay? So here's some chymographs just showing you that we measured some elongation rates, and this like 5-fold increase that we saw before, it's maintained. We can also do this cool anisotropy trick that I love so much now that we have this fluorescent prophyland. And here's something that we weren't expecting to find, but we're excited about. And I guess I didn't put it on there, so sorry. So it turns out that prophyland can bind dimers of tubulin, because we did these assays where at a temperature and concentration where tubulin couldn't polymerize. And basically the KD that we get is around 1.5 micromolar, which is still very high. But the amount of prophyland that we, the KD that we thought that we measured for the sides of microtubules is actually 10 micromolar. So I actually have this idea. It seems very attractive to me that if you bind the subunits of tubulin that are going to get added on, that you have this way to sort of somehow facilitate that more efficiently. So that's what's happening on the tubulin front. And we can also look and see where the prophyland is with the fluorescent tag. So here I'm showing you the microtubules in black. Here's the M-apple channel. If you look carefully, you can see that there's things that look like microtubules in there. And down here's the merge. So spoiler alert, it binds the sides. And here's just a movie showing you the dynamics. Eventually a microtubule is going to start to polymerize, and you'll notice that the M-apple prophyland in the M-apple prophyland channel that just shows that looks like a microtubule. And if you do line scans over some of these microtubules, you can actually see that prophyland seems to be dynamically associated with the lattice, and it's diffusing around as far as we can tell, or it's coming on and off. I don't know. It's really fast. So it's something that maybe we should follow up on, but there's that. Okay. So when I was a graduate student in a plant lab, and we tried to tag proteins and put them back in plants, my PhD advisor, said, well, Jess, if you're gonna do that, the gold standard is to see if it rescues a knockout phenotype. And now we have this great tool in mammalian cells, CRISPR-Cas9. So we used neuroblastoma 2A cells to make our knockout. And this is just what these cells look like if they're undifferentiated or differentiated. Granted, our lab is super interested in neurodegeneration. I don't know if we necessarily want to go there with primary cells. So there's a lot of groups that use these cells in that context. So that's very attractive. It only has 2 copies of the genes, which is great for if you're trying to CRISPR things. They can differentiate into things that look sort of like neurons. Anyway, they're kind of cool cells, except they kind of suck for all the things that we would want to do to test them, like a migration assay or cell division. It takes them a really long time to divide. Who knew? Anyway, so what we did was, here's a Western blot just to show you endogenous levels of prophyland that our knockout line, we actually have 2 different clonal knockout lines, doesn't have any of our prophyland there. And then when we rescue it, there it is. And we also can rescue with our M-Apple version. And I also made a halo tagged version, which we'll get to in a second. And both of them, you know, can rescue this pretty well. Here's a Kumasi of our blot to show you that we loaded things equally. And we also used tubulin just to show that something about the loading as well. And these cells have a phenotype. Both of our knockout lines, there's always fewer when you go to passage them. And I think that others have reported that they get stuck in the cell cycle and eventually they resolve that issue. But like part of the reason there's less is they take a lot longer to divide on top of the already slowness of these cells. Okay, so because we have purified protein and microscopes where we can figure out how the volume of a cell, we did some quantitative Westerns where we loaded amounts of tagged prophyland and apple tagged or our untagged version. And we kind of calculated how much prophyland was in a cell. So in these cells, normally there's 121 micromolar. And then if we rescue with not a tagged version or the M-Apple tagged version or the halo, depending on when we've done this 4 times, you know, these are not statistically significant from the amount of endogenous that we get. So it seems that we're able to rescue it depending on the day close to endogenous levels. And what can we do with that? So I told you these cells are difficult. So what we came up with was let's plate them on micro patterns. So then they're forced to have the same shape and then align the images on top of each other to see if there's a departure to actin or to microtubules or just the cell shape in general. So what I'm showing you here is microtubules only because it looks prettier. But you can see for endogenous, not tagged, tagged, or even the knockout, that's what the cells look like. Here's what it looks like when you align those on top of each other. And we can do this for different days where, you know, on a given day, you know, this is the whatever our endogenous is normalized to 1, and the departure from that is for the other tags. Okay, so we tried to quantify it that way. Looks pretty good. Here's where it gets really cool. Okay, so here's the new stuff. We're gonna drop the update to our preprint soon. So that's where our preprint ended. And we had this really cool thing where we were like, oh, we've got these tags, like we have this opportunity. Can we visualize what propyllen's really doing in a cell? I mean, it was one of those moments where you submit the paper and you're like, duh, of course, you got to go do that. So what we did was we titrated our halo ligand to see less of the amount of propyllen that's in a cell, okay? And this is how much ligand there is. But just to remind you, because there's so much propyllen in a cell, at 2 micromolar of our ligand, you're looking at about 1.6% of all the propyllen that's there, okay, in the cell. So very little. So we titrate this way down. You can see at the higher concentrations, it's everywhere, including in the nucleus. At a lower amount, you can start to see maybe single molecules still in the nucleus. At about 100 nanomolar, we can see something that looks cytoplasmic. Maybe at 10 nanomolar, we see things. And there's actually some in 1 nanomolar. They're just really hard to see, okay? That's the actin and microtubules in those cells. So I don't know we haven't quite figured out how to quantify this, but if I look at that, I think like, okay, maybe it's doing its thing with actin monomers. Okay, except if we take this concentration and look throughout our dish, sometimes this is what we see. This is the prophyllin channel. This is the actin and microtubule channel. And what you'll notice is this looks like microtubules, which I was like, that was exciting for me. So I went and just overexpressed prophyllin because these are all rescued versions, right? And probably not surprisingly, that didn't do very much because we're overexpressing over endogenous, and the endogenous doesn't have the tag. So we couldn't really, you know, we probably could get it if we titrated everything again, but you know, we tried. It was an idea. Okay, so if I want to see pools of prophyllin, though, we have to take advantage of some of the mutants, okay? And I promise I'm almost done. So just to remind you, R88E is a mutant that doesn't bind actin. It's over here. G118V is really cool. This one is one of the ALS mutants, and it doesn't bind microtubules. It's over there. And then Y6D is used usually for disengaging polyl-proline. So here are some ideas of what I predict would happen. I don't know if I would have predicted actually that Y60 would go to microtubules, but that's where it's going to go. Oops, sorry. So if we look at this, what you see is in wild type, we see the speckles of our prophyland hanging around. I'm not sure if it's actin or microtubules. It's probably actin because we can't resolve those individual monomers. If we look at R88E, which doesn't bind actin, it pushes the prophyland towards the microtubules. So that's cool. G118V, it doesn't bind microtubules. We see something that's a little bit more diffuse. We also see these aggregates, which I could have stressed out the cells, but it's also something that you can see with that mutation for prophyland as part of the disease ALS. And then Y6D, it doesn't bind polyl-proline, but that seems to be shifting towards the microtubules as well. And I don't know, I haven't fully thought that through, but it probably has to do with what it's doing with formin. And just to give you a hint at where we're going, we also can look at these things in live cells. So I'm showing you actin and microtubules, the same things as before. And interestingly, if you look at our R88E, which doesn't bind, there's a little bit of bleaching. We're still working it out. You can see that the prophyland seems to be diffusing along the lattice in the microtubules in those cells. Of course, not the one I was trying to image, but it's there. So I'm pretty excited that we can see that, and hopefully we'll come up with something quantitative to look at that soon. I'm going to blow through the ALS stuff, but basically this is my plea to say that anyone who's not a cytoskeleton person, you're really studying the cytoskeleton, sorry, especially in ALS, because not a lot of people are looking at the cytoskeleton in ALS, and they're only kind of looking in the context of what's happening to the transport or the tracks. So I think that there's a lot we can learn from the about 8 genes that are cytoskeletal that are associated usually with microtubules for ALS, but there's 8 variants. And just as a teaser on the micropatterns, here's our knockout, here's a rescue, here's one of the ALS mutants. So a savvy person might say that is not fully rescuing. Absolutely. I don't know why yet, but we would love to use our prophyland to figure out if the dynamics are somehow different there. So that's where we're hoping to go. Here's the main takeaways. We think that we have a decent tagged prophyland that can help us learn some stuff in vitro and in cells. And with that, I'll take any questions if anyone has any.
Thank you so much, Jessica, for such a wonderful talk. Maybe I'll start with the questions while other questions trickle down. I was wondering where you were showing your fluorescence anisotropy, and this is more like technical question. You were looking at the anisotropy of prophyland itself, right?
Okay, so for the very last one I showed, yes, but for the others,
but in the beginning, yeah.
In the beginning, it was actually GFP thymosin beta 4 was the fluorescent thing that was in there. So that gives actin and prophyland, hopefully nothing to interfere with their binding for each other.
Okay, so it was used some sort of like a not really tagging prophyland. Like I didn't understand how it was used to measure prophyland's activity.
So it's sort of a biochemistry trick where the idea is thymosin beta 4, GFP thymosin beta 4 is going to be bound to the actin, but at some point it's going to come off. It has an off rate. Okay. So when that happens, that gives prophyland a chance to bind. So it will change how it's spinning in the solution, and then you can detect it.
But that should have changed, the polymerization for actin, right? Like when you provide more and more proliferant. So how did that not change the anisotropy in the untagged version?
So in that one, the actin's not polymerizing. It's staying monomer. We're keeping it at very low concentration. Yeah.
Okay. Thank you so much.
Yeah.
Right, great. We have a question from Isabel Palacios, wondering if in a wild type situation, is the prophyland acting on both actin and on microtubules all the time over all the cells, or is it regulated? Are actin microtubules competing for its function?
Okay.
Yeah.
So in a cell is a little bit tricky. So by KDs alone, actin should win, but in a mammalian cell, there's thymosin beta 4. So there has to be a free pool of prophyland that's probably not just bound to actin monomers. And I mean, we're getting closer, but we still can't visualize that. So in theory, they're competing. If we ignore everything else that's in the cell except for actin, microtubules, and prophyland, actin wins. But we don't really know how the other things in a cell that are interacting with actin or even with prophyland are kind of soaking up the sponge. So we're not sure which way it's going. But I do think it is competing because we don't, in our wild type case, we don't always see the tagged prophyland on microtubules, only some of the cells. I don't know why those cells, it could be an expression thing. But I think it is possible that they're competing at the same time. It could be something that the cell's doing in that moment. I don't know. Does that help? I hope it does. I hope it does.
Thank you, Jessica. I think Michael Shannon has a question. Please go ahead and ask your question.
Hey, yeah, first of all, thanks for the talk. That was the way you described all of these controls for, you know, what an FP and a linker does to a protein is where my head's at all the time. So thanks for that. But I wanted to ask, have you ever heard of these alpha tags and or these V5 tags? Are you interested in them?
I think we actually have a version of prophyland somewhere in the lab that, you know, I collected from somewhere that has the V5 tag for like using for like antibody labeling, I think.
That's right.
Or like pulling it down. So I think that they're fine. I think, and many people have used like different tagged versions, you know, so cautionary tale. You should always go and test if your tag is somehow affecting your protein's activity. But sometimes, you know, we all have vectors that we just throw the protein in, and it might work good enough to say at least, you know, it does interact, but maybe our KDs aren't right or something like that, right? So yeah. I think it's okay. I think for now knowing what I know, I would go back and try like just adding the little linker to it and see if it changes anything. And maybe it doesn't, but at least, you know, I'll know that in our system with the things that we tried, how it behaved. Yeah.
Yeah. Could if I was to, do I have a second just to follow up on that?
Yeah, please go ahead.
Well, the reason I was asking was because you have these amazing images of overexpression in a rescue and in where the endogenous is there still. And I mean, you're titrating down to that single particle tracking with the Janelia floor dyes, aren't you? I mean, my thinking with my molecule is that if I can get one of these little alpha tags into the endogenous with CRISPR, and then add in using lentivirus or something, the nanobody with a halo tag that then binds the Janelia floor dye. Wouldn't that be cool for your prophyland situation? That was what came to mind.
Yeah, totally. So we have like Storm even, like we just didn't do it, you know, just to sort of exactly like to really get there. For us, we're not doing Storm in the way that you can do it in a live cell, but like if we wanted to just resolve like, hey, is it in there? And for some of the stuff that we're interested with ALS, I think that it will start to become relevant. And you're right. I think that that's a really good approach. And I just totally didn't realize that there were these, like so many of these smaller tags. So we might go back and, you know, yeah. But that was what was great about this like ASAP bio thing. Like as a new investigator, it's wonderful to get feedback. Sometimes you don't always know, like, should I send this to random people in the field or whatever? But it was really nice to get that feedback. It was a lot of feedback, but it was very helpful.
Cool. Thanks a lot, Jess.
Yeah.
I'm glad that
I love that.
Sorry, go ahead.
Yeah, no, both Ankita and I are big fans of open source biology. So I was just wondering, I mean, one thing that I am always struck by with those micro patterns, and it looks like maybe you were using the ones from like the cytomorpho lab, right? Where we've got triangles and T's and circles. You know, when you put cells on there and you see the way the actin shows up at the corners where these points of high stress, you showed us all triangle patterns. When you use others, you know, circle versus, you know, do you see any localization of prophyland like towards or away from sort of, you know, different, I guess, like shape inflection points, you know, sharp versus smooth, that kind of thing?
So we would love to do that experiment. We have not done it. Basically, we tried a whole bunch of things like patterns, and these were the ones that our cells happen to stick to because unfortunately, N2A cells are not part of their normal catalog. But now that we sort of have an idea of how to get some of them to stick down, I think we could go back and do that, and that would be really cool. Yeah.
Yeah, no, because you know, I'm always just thinking about cell shape and how is cell shape affecting, as a causal piece of this. And I was wondering how prophyland is gonna respond. And I also, following up on one of the earlier questions that we had, it's really, and as somebody who doesn't really think about biochemistry very often, I think about cells and about tissues. It's really hard to get your head around how something that's so ubiquitous can be doing anything specific at all, because it's like freaking everywhere and it interacts with everything and it's doing so much. And I don't know, it's not really a question. I'm just sort of expressing
so important that it needs to be
at
everywhere. And they just say, you know what, I'm just going to put prophyland everywhere. So whenever we need it, we have it. Maybe.
Okay, is that, so you think it's sort of, it's like it's on reserve and it can act where it's needed?
I think I'm sort of thinking of it in a different way. Normally people are thinking of prophyland as it's keeping the actin, so it's on reserve, so it can be ready any moment it's needed, which maybe, that's probably right. But for me, it's like, just remember that there's still free prophyland out there and who knows what that's doing. Or maybe there isn't and it is bound to the microtubules or something else, but yeah.
So follow up of that, the R88, and now I forgot what the whole thing, the mutant's name was. That was the actin mutant, right? But in your images, I could see between the control and the mutant that the microtubules have like severe effect, like they become very close looped, right? So do you think it's directly doing something? And I guess, it's coming back to the other question we had about whether, it's a direct impact on microtubule stability of some sort, or because that mutant should be more specific to actin.
It's true. So I think that mutant maybe isn't as perfect as not always totally not binding actin, just as a spoiler alert from some other assays that we've done. So that might be part of it. But yeah, it might be doing something with the microtubules. We haven't gotten our EB1 out to check microtubule dynamics with these yet. Maybe that's something we'll try to do in the future. And I think now that we can see the prophyland, at least somewhat on microtubules, are sort of figured out how to like get that more consistent. We can start to like address that question, like, does it get enriched when we see more of these microtubules? Are they more bundled when we can, I think those are good thoughts.
Thank you so much. I think am I missing something in YouTube? I know. Okay.
Yeah, I think we're good there. I just wanted to say thanks, John Cooper, for your points in the chat about prophyland functioning all the time to keep the actin system turning over. That seems like an interesting way to think about it. And Jessica, if you want to comment on that, you can feel free.
No, he's right. I totally ignored something else that prophyland does. So I think that comes up to his point a little bit too.
Okay. Yeah. This is why I think about cells, not molecules. What you do is too hard. Awesome. All right. So yeah, if we don't have any more questions, we can go ahead and wrap up. So thank you so much, Laurel and Jessica, both of you, for really different talks on really different scales, but both fantastic for thinking about cell migration. So I'll go ahead and I'll end the live stream here.